So on to the main event here. It's my great pleasure to introduce Dr. David Baker of the University of Washington. At QB3, we work with academic researchers and entrepreneurs who are studying natural systems and developing new technologies in biochemistry and biotechnology. And many are investigating or developing new therapies. On the chemical side, usual process involves searching enormous natural libraries of compounds from leads, but there is an emerging field of rational drug design. Whereas when it comes to major macromolecules, rational design is extremely daunting because of the enormous complexity involved in protein folding, both predicting what a long chain of peptide residues might fold into, or trying to figure out how to design a chain of peptides that folds into the shape you want it to. So today we're extremely lucky to hear from the leading figure in the new field of Denovo protein design, David Baker, director of the Institute for Protein Design at UW. He's going to tell us how he and his team are designing custom proteins from the ground up. And just as importantly, hopefully he's going to tell us why we want to do so, possibly to break free from limits that have been imposed by life itself through billions of years of evolution. He's got an extremely long bio, but it's massively impressive. So let me just read it for you. He's the director of the Institute for Protein Design, an HHMI investigator, Henrietta and Aubrey Davis endowed professor in biochemistry, and an adjunct professor of genome sciences, bioengineering, chemical engineering, computer science, and physics at the University of Washington. And his research group is focused on the design of macromolecular structures and functions. He received his PhD in biochemistry with Randy Sheckman at Berkeley, and did his postdoc with David Agard at UCSF. This is where I get to brag a little and say that David Agard was QB3's first director at UCSF, and Randy Sheckman is also a QB3 faculty affiliate, so a little reflected glory. Dr. Baker has received awards from the NSF, the Beckman Foundation, and the Packard Foundation. He's a recipient of the Breakthrough Prize in Life Sciences, Irving Segal and Hans Neurath Awards from the Protein Society. Many other prizes. 65 mentees have gone on to independent faculty jobs, been published over 500 papers, 100 patents, and co-founded 11 companies. So these technologies are becoming real and will have impact on patients in the near future. Dr. Baker, a great pleasure.
All right, thanks. Can you see my screen okay?
Yes.
All right. So yeah, thanks for the invitation and introduction. So I'm going to focus on de novo protein design today and talk a little bit about structure prediction at the end. So the basic picture, which has driven our work for many years, actually up until about a year ago, is the one on this slide. So and the basic idea is that protein structures are determined by their amino acid sequences, and the sequences encode structures by virtue of the structure being the lowest energy state for the sequence. So if you want to predict structure from sequence, you search through the different possible states that the protein chain could have, searching for the lowest energy one. And if you want to make up brand new structures, you come up with a new structure, and then you find a sequence whose lowest energy state is that structure. And over the years, we and our colleagues have developed a computer program called Rosetta, which calculates energies and does the sampling through different possible structures and different possible sequences required for this. Now, as all of you are aware, deep learning is starting to have more and more of an impact on science. And I'll show you towards the end of my talk how deep learning is sort of transforming, or at least giving a different perspective from which to approach both of these problems. So I'm going to focus today on de novo protein design. This is an interesting problem because the number of possible proteins is absolutely enormous. For a 100 residue protein, they're on the order of 10 to the 130th sequences. And nature has only sampled a tiny fraction of these. So everything I'm going to be telling you about today are completely brand new proteins made up that don't have any sequence similarity to naturally occurring proteins. And I'll illustrate these methods in the context of what we've been doing in the last year and a half in response to COVID. So when the genome was released, we built models of the spike protein, which turned out to be quite accurate, and then switched to using cryo-EM and crystal structures as those became available. The first thing I'll tell you about is the design of antivirals. So here the idea, I think you all know that the spike protein of the coronavirus interacts with ACE2, the ACE2 protein on the surface of our cells. And the basic idea was to build proteins which bind to the ACE2 binding site on the receptor binding domain of the virus and prevent that interaction. And the first way we tried to do this was to take part of ACE2 and then to build small proteins around it. The second way we tried to do this was to generate very large numbers of completely new small proteins, backbones, knock them against the structure, and then design the interface for high affinity binding. And we actually were able to get binders using both of these approaches. It turned out that the binders that we got using the second approach, the completely de novo approach, where we didn't use anything from ACE2 or any natural occurring protein, were much, much higher affinity. And the reason is probably that we're much freer to come up with sort of optimal binding modes if we aren't restricted to some feature that's present in a naturally occurring structure. So here are examples of binding proteins that we've made using both of these approaches. And so this is one of the de novo ones. And you see it's got some remarkable properties. It's just 55 amino acids. It's stable at 95 degrees, at above 95 degrees C. And it binds with KD below 100 picomolar to the virus, just a single copy. So this is a cryo-EM structure solved in the Wiesler lab. I should say that this protein was designed by Longjing Cao, who's really the hero of this first part of my talk, along with Brian Coventry. And here's a structure of this little protein bound to the spike trimer and by cryo-EM, and you can see 3 little proteins bound. And this is now a comparison of the computer model to the cryo-EM structure. And so you can see that the design protein is exactly where it was supposed to be. And if we look at the side chains, you can see that those are exactly where they are supposed to be as well. So Longjing and Brian designed this protein basically completely from scratch, just given the structure of the protein, to bind very precisely to this part of the spike protein. So this protein is very potently neutralizes the virus with a IC50 of 15 picomolar, which is 0.15 nanogram per mil, so super potent. And it protects both hamsters and mice from the coronavirus. This is now just being administered intranasally by spraying into the nose. So these are hamsters that got this protein from 48 hours before to 24 hours after infection. And you can see that they're essentially, by the end, they're kind of identical to animals that didn't even get the virus at all. So to get still higher avidity and resistance to escape, we took advantage of the fact that the 3 of these proteins bind to the spike protein simultaneously, and we built symmetry matched design proteins that are now trimeric versions of the binder. You can see the cryo-EM structure here. So this is the spike trimer down here, and this is the design trimer. And this protein binds with an affinity that we can't measure to the virus because there's now 3 times three copies. And it's very potent in neutralizing, basically every strain of the virus that we've seen. These are ACE2 mouse data. And so this is the South Africa and UK variants. I don't remember all the numbers, but it's the single compound seems to be quite, is quite escape resistant. And that's again, because we have so much avidity. And finally, these proteins have some really nice properties as potential drugs. The cost of goods for making them is far lower than an antibody. So this is just a lane expressing. This is a lane SDS gel lane for cells expressing the protein, and you can see it's expressed at very, very high levels in E. coli. If you just boil the E. coli, most of the E. coli proteins are insoluble after you spin, and what you have left in the supernatant is primarily the design protein. And So the cost of goods, it should be extremely low. And so this is currently advancing towards clinical trials. And as I said, in all the animal tests that we've done, these trivalent versions, and we have several different types, ones that have 3 different domains strung together and ones that are symmetric trimers, protect against all the strains that we've tested against. So we're quite excited about that. And unlike antibodies, they don't require a cold chain and so forth. So Longjin and Brian have used this approach to design binders to quite a number of protein targets, both pathogens. For example, here's a binder that binds to both group 1 and group 2 influenza hemagglutinins for basically a similar approach for flu therapeutics, as well as binders to different growth factor receptors, cytokine receptors. And in all these cases, all that they're starting with is a structure of the target. So basically what Longjin and Brian have come close to solving is the very general problem of, given the structure of a target, design a small protein which binds with high affinity and specificity to that target. And this is just an example of the specificity data. These are the protein targets. These are the binders. These are what they look like on their surfaces. So you can see they have very different shapes and electrostatic potentials. And you can see they're quite specific. So here's the coronavirus binder I showed you before. It does not bind to any of the other targets and so forth. Here we have 2 different binders to 2 different parts of the EGF receptor. Here we have crystal structures of a number of these binders. This is one that was solved in Ian Wilson's lab recently, of this general binder to group 1 and group 2 influenza hemagglutinins. And you can see again, the binding mode is almost exactly as designed. And these are some of the other structures, for example, for the FGF receptor, the NGF receptor, and the IL-7 receptor. And so we've been making, this method works quite well. So we've been making binders to many different cell surface receptor subunits, as well as viruses. What we're really excited about doing is making a whole series of novel and biased agonists using these. And basically, since they're very small proteins, we can array them both as dimers, shown here, or in other types of configurations, and elicit different types of signaling responses. So just to illustrate this concept, this is a collaboration with Chris Garcia from a couple of years ago with the EPO receptor, where we showed that basically, depending on the geometry with which EPO receptor subunits are positioned, rather different signaling responses can be elicited. Now with these binders to all these different receptor subunits, we can make a really exciting new class of ligands that bring not only different numbers of copies of the same receptor subunit together, but we can actually start mixing different receptor subunits. So we're very excited about being able to elicit cell fate transformations with these synthetic ligands that aren't possible with the natural ligands. So what I've talked about so far is the design of very rigid proteins that bind to specific target surfaces. It's kind of like designing locks for keys for locks. Now I'm going to talk about design of proteins that can move. Here's a system, a molecular device we've designed, which has an off state and an on state. And the opening of this switch is driven by binding to a target, in this case, the coronavirus again. In the open state, this device emits light. It luciferases reconstituted. And this works really well. This is basically the sensor. It's dark in the absence of the virus. The virus is added, and we get this rapid increase in the amount of light emitted. And our collaborators in the Silk Lab at Tufts have put this sensor onto masks and gloves and gotten these really cool results. So we're quite excited about the potential of these sensors. We can also use these sensors to detect the amount of the extent of protection that an individual has from vaccination. The basic idea is that the sensor is under thermodynamic equilibrium. So if antibodies are added, if you have antibodies in your serum that bind to the RBD, they will pull away the target from the sensor and it will close. And you can see both in mice and in humans, the amount of signal change correlates with the amount of neutralizing antibody. And finally, we've been able to build these devices for for quite a variety of different targets. And so it's a very general type of system. And actually, each of those little design binders that we can now make can be inserted into this type of sensor to generate responses. And so, for example, this is a troponin sensor, which only emits light in the presence of troponin, whereas none of the other sensors that we've made to these other targets open in the responses to troponin. And we can also use this sort of design of multi-state system here to for a rather different challenge. For example, in this case, distinguishing tumor cells from normal cells, and in particular doing logic operations on the surface of cells that allow distinguishing cells that have two markers, a marker 1 and marker 2, from cells that have just marker 1 or marker 2. And so the basic idea is if cells just get one component or the other component, the sensor stays closed, the switch stays closed. But if both markers are recruited, then the sensor opens and we get recruitment of an effector. And I'll illustrate that here in the context of CAR T cells. So here we have a target cell that has 3 markers on its surface, two tumor markers and a healthy cell marker. So we want to kill the cell only if it has the 2 tumor cell markers, but not the healthy cell marker. And the way we do that is we target to the 2 tumor cell markers the components of the switch that I just described, and we target to the healthy cell marker a component that sequesters, that binds to one of the 2 components of the switch more tightly, and so basically prevents it from opening. And you can see this works really well. Cells that have the 2 tumor cell markers lead to activation of the CAR T cells and eventually killing of the tumor cells, whereas cells that have all 3 are not touched. So this could really help with specific gene targeting, not only for CAR T cells, but for gene delivery and other applications where you really want to target subsets of cells. And the final area I'll touch on briefly is vaccines. We've been developing these general methods for designing self-assembling nanoparticles. So these are made out of 2 protein components, in this case, a purple and a gray one, and they are placed on the symmetry axes of a regular polyhedron, in this case, an icosahedron, and then you can rotate them back and forth and up and down, and then design interfaces between them. And when we make these 2 proteins in E. coli, they're very homogeneous, and we've designed probably on the order of hundreds of different particles for different purposes thus far. So my colleague Neil King at the Institute for Protein Design, my next door neighbor, has taken these nanoparticles and he has placed fused to their surface, the receptor binding domain of the spike protein. And this construct looks like this. And here are pictures of these particles. Now this compound, this immunogen turns out to elicit extremely strong responses to the neutralizing responses to the virus, much stronger than the trimer, which is of course the basis of the mRNA vaccines. And this is currently in phase 1/2 clinical trials, and it's now has funding to move into phase 3. So hopefully this one will make it out into the world in not too long. The responses in animals have been tremendous. So the cages I've described so far, as I said, were made out of 2 protein components, and then Neil fused to the surface of the virus the receptor binding domain of the spike protein. But we reasoned that if we could build the cages out of proteins that were themselves functional, we could unite form and function and not require a subsequent fusion step. And the protein we decided to test this out with is our antibodies. And the reason is, as you all know, antibodies are really the workhorse of biotechnology. They're the basis of many therapeutics, and they also have C2 symmetry. So in this case, here we have a protein that has a design protein of 5-fold symmetry that is placed on the 5-fold symmetry axis of an icosahedron. And here we have the antibody with C2 symmetry on the C2 axis. Here we have instead an octahedron with a design protein on the C4 axis and the antibody again on the C2 axis. And this generates quite a variety of different types of nanoparticles with different numbers of antibodies. So this icosahedral one has 30 antibodies sticking out in different directions. This one has 24, this one has 12, and here's one that just has 2 antibodies sticking in 2 different directions, so just 2. And what we found, and this is work by Robbie Devine, what we found is that we can take one antibody, just add one of these nanoparticle forming proteins, and we can generate these different types of structures and then add them to cells. So this is an antibody against DR5, one of the death receptors. The antibody itself does not do too much to cancer cells, but when it's built, it's assembled into one of these nanoparticles, we get pronounced tumor cell killing. What we found is that this is quite a general result. If we take an antibody that's only weakly agonistic or not agonistic at all, and we build it, we assemble it into a nanoparticle, we can get pronounced activation. So for example, for T cell and B cell activation, among others. And so we're very excited about this now as a general platform for turning antibodies into agonists. The nice thing about this is it doesn't require any modification of the antibody. You just get just make the assembler protein, you mix it with the antibody, and then you have these beautiful nanoparticles. So on the topic of self-assembly, in addition to making these closed structures, we can also make open structures like hexagonal sheets. And here I'm showing a hexagonal lattice that's made out of a designed C3 symmetric protein and designed C2 symmetric protein. And when you mix them together, when they're expressed in E. coli, they form this hexagonal lattice. And if the 2 proteins are purified and then mixed together, you form this really, really extensive hexagonal lattice shown here. And we can fuse to the trimeric component, or actually to both components, binding domains for cell surface receptors. And what we found, and so it becomes a really interesting way of doing cell biology, because you can form these sheets on the surface of cell. So we can activate TCR signaling, for example. We can activate the angiopoietin pathway. And what's quite interesting is that you can actually completely shut down endocytosis, because the membrane can't deform against the tension of this layer. So everything I've talked about so far has been soluble proteins, but we've also been designing membrane proteins. And what I'm showing here is a designed transmembrane ion channel. This is it's got 2 rings of helices and a central channel through which ions can pass. And what's quite remarkable is this protein turns out to be a very selective potassium channel. You just look at the selectivity over potassium over sodium. And this is quite interesting because we actually did not design it specifically for potassium. This was just an emergent property. And so now what we're doing is we're changing the lining of this channel to probe how the composition of the pore affects conductivity. So that protein was made out of, I'm sorry, I left out the picture. It was 2 concentric rings of alpha helices. It was a hexamer of helical hairpins. So there's of course another class of designed of membrane proteins in nature, which have been much more difficult to study, and those are out-of-membrane Betabarrels. And Anastasia Vorobyeva figured out how to actually design new Betabarrels from scratch that insert into membranes. It was really quite interesting. The trick was to actually substantially reduce the beta sheet formation tendency of the strands, because the first design she made all tended to aggregate horribly. And it was by weakening the propensity so that they would only form beta structure after inserting in the membrane that she was able to get this to work. And Samuel Burhanu-Lema has gone on to use these methods to design larger Betabarrels that now have substantial pores in it. This is comparing the design model to a crystal structure. And this is very exciting because, as you know, transmembrane Betabarrels are used in many applications, for example, for single molecule DNA sequencing. That's all just adapting naturally occurring channels, but now we can build new ones from scratch. And we think there's truly an almost endless number of applications of these to sequencing and beyond. And the final example I'll give you of protein design is work from Alexei Corbet. And so he has been designing rotational protein machines. And so he designs protein rings shown here and axles shown here. And he's worked out ways of getting these rings to assemble on the axles, as shown here. And he's made quite a few different systems. So here's one where the axle and the rotor both have C3 symmetry. And so as you'd expect, the rotational energy landscape has 3 pronounced minima. And this is a structure that Alexey has determined. The other systems here do not have a symmetry mismatch. So for example, here's one where there's a D3 axle and the rotor has C5 symmetry. And so in that case, the rotational energy landscape is much flatter because there aren't these 3 minima to fall into. And you can see we get much fuzzier density. The one I'll focus on a little bit more is this one. This is a D8 axle on which Alexey has designed interactions with 2 C4 rings. And he's collected a lot of cryo-EM data on this system. And what's neat is he can distinguish different rotational states. So here is one where the 2 rotors are eclipsed. They're kind of these arms are pointing in the same direction. And here's one where they're staggered. And so this is kind of a neat system. So we've been able to design a system where there's sort of one-dimensional rotational diffusion. And as you can imagine, Alexey is now building in catalytic sites at the interface between the rotor and the axle to drive, to try and improve, generate directional motion by using the chemical fuel. Okay, so now that's basically sort of a kind of a whirlwind tour through what we can do with Rosetta. And now I'm going to show you a couple other ways that we can design proteins. First of all, for a number of years, we've been trying to enlist the general public, or have been enlisting the general public, in protein structure prediction and design through a game we've developed with collaborators and colleagues called Foldit. And here, game players start with an extended chain and they can bend it up and twist it up to make all types of structures. And Brian Kovnik, who's really the genius behind this project, has made a bunch of these proteins and solved structures. And it turns out that non-scientists can now design proteins using this approach. And so now I'm going to switch over to deep learning, which has been quite a lot of fun in the last year and a half or so. So we first started off thinking about deep learning methods for protein structure prediction. And so we developed methods for taking amino acid sequences, predicting distances between the amino acids, and then building those into 3-dimensional structures. And we developed a method called TR Rosetta, which does this. We realized after we had this that we could turn this around. And the analogy is, you're probably familiar with methods that take, say, many images of cats off the internet and build classifiers that can recognize cats and distinguish them from other objects, and then use those networks to hallucinate brand new images of cats that are images that the network thinks are cats, but are not the image of any particular cat. We can do exactly the same thing here. We can feed the network random amino acid sequences. They won't be predicted to have any particular structure, but then we can go back and optimize the sequence to have a strongly predicted structure. And that's illustrated here. So we have these random sequences we start with. They have very fuzzy distance maps. And then we optimize the sequence, simply favoring sequences that are predicted to have strongly predicted, that have sequences that have strongly predicted structures, like look like some cat, but we don't care what cat it is. And we get these different types of contact maps out that correspond to different types of structures. And we've gone into the lab and made a bunch of these proteins, and they actually fold up into stable structures. And we've solved structures, crystal structures and NMR structures of a bunch of them, and they're quite close to the design models. So this kind of purely deep learning method can now generate brand new types of structures. So what we've been doing with this, I'm not going to show you because I don't have time, that this method can actually model whole energy landscapes because it's actually predicting probability distributions over possible states, which is something that's hard to do with an energy-based model using Rosetta. So what we can do to design functional proteins is we can take some small part of the structure and fix it of a functional protein, and then just basically hallucinate the rest. And so you're basically saying to the network, okay, keep these parts fixed, and then make the rest whatever you want, but just something that looks like it'll fold. It's like giving a network the ears of a cat and saying, build build a cap that fits these ears. And so we get, so we can design proteins in this way that scaffold various types of functional sites. So this C3d mimics that mimic where C3d functions in the complement cascade. Here are mimics of insulin. So anyway, that's fine. So, but there's kind of the most recent development is many of you are probably aware that DeepMind made considerable advances in protein structure prediction last November. But they didn't really explain how they did it, and they didn't make the method available. And so, since we've been interested in protein structure prediction and protein design, we thought we'd basically try and use. They presented a 10-minute talk. So there were a few clues. So Ming-Kyung Baek, who's a total genius, managed to basically create something which a network that actually is somewhat different from what they use. So it has basically processes one-dimensional sequence information, two-dimensional contact map information, and three-dimensional coordinates all in parallel, and outputs a final structure. And this method really works well. So we've set up a server, and this runs on the PDB every week, and it's quite a bit better than other available methods. We've run on cast targets in server mode, where it's just taking a few minutes per target, and it's still not quite as good as AlphaFold, the DeepMind method, but AlphaFold the structures were predicted over quite a number of days on many GPUs. So it was a much more intensive search. So the exciting thing about these models is they turn out to be really good for solving molecular replacement problems and cryo-EM problems. So here are a couple examples of structures that were solved by a molecular replacement in A and B. And here's one that was used to solve a cryo-EM map, where basically no other methods that were available worked. And so I first sent out emails to everyone I knew asking if they had problems that we could help solve, and actually a number of them were solved. And then we more recently set up a server, and I get emails every day saying, oh, it's great, we solved our, we had this molecular replacement problem around for the past year that we couldn't solve, and now we've solved it. So it's really exciting. So this is now on a server. We've also released the source code, and so people can build on it and use it. So we're really exciting. This is not quite perfect structure prediction from sequence, but it's getting close enough for many applications. So Ming-Kyung realized that she could go further. This not only will model the structures of monomers, but you can give it the sequences of multiple different chains, and it will generate models of complexes. And so this is a kind of fun example. This is the full IL-12 receptor complex, consisting of 2 IL-12 subunits and 2 IL-12 receptor subunits. Chris Garcia's lab had solved the structure, but hadn't been able to really model into it. And Ming-Kyung's built a model of it using this method, and it fits the density really nicely. So this is not quite available on the server yet. So if you want to build models of complexes, just let me know. And we're starting to build models of proteins of currently unknown structure, like GPCRs. Here's a model of the human sweet receptor. So if you have complex structures of biological interest you are interested in, let me know, or try out the server. Yeah, so we're really excited about this. We're moving on to now try to build models of protein-protein complexes on a much larger scale. I'm a little worried about time. So, whoops, I pasted things in the wrong place. Shoot, my acknowledgement slide is, oh my God, where is that? Okay, well, these are the people who did that. This is the most important part. Oh no. So Ivan and Sergey developed TR Rosetta. Sam and Temuka did the experimental testing. Chris and Basil did the landscape mapping. I didn't really have a chance to talk to you about. Doug and Sydney and Ju did the constrained hallucination. I mentioned Ming-Kyung Baek, who's just did an amazing, it was an amazing virtuoso performance with the Rosetta TT Fold. And I am just going to have to find my acknowledgement slide, because this is really horrible. I don't know where it went. I don't know what happened. You can see what you were spared from. Okay, well, I'm just going to go. I'm sorry to put you through this, but I am going to just tell you who the stars of this were. So the design binder, that's just amazing work by Long Jin Kao and Brian Coventry, who I think pretty much solved the problem of designing binders to arbitrary protein structures. Then we've had many outstanding collaborators for all the cell culture and in vivo experiments. The biosensor work was done by Alfredo Rubio and NDEA. The logic for detecting tumor cells work was done by Gillian Bruffy, Mark Lajoie, and Scott Boykin. I talked about Neil King's lab work. He's a whole host of collaborators on the coronavirus vaccine. Robbie Devine designed the antibody nanocages. The hexagonal sheets work was done by Ariel Ben Sasson. The potassium channel work is done by Chunfu Zhu and Peilong Liu. And the transmembrane beta-barrels, I mentioned Anastasia Vorobyeva and Samuel Perhanyu, and Alexei Corbet designed the molecular machines. So sorry about that. That was a very bad omission, and I'd be happy to take any questions.
Thanks for an amazing talk, Dr. Baker. So yeah, you can submit your questions in the Q&A field down to the right of your window. Would you like to read those yourself, or would you like me to read them, Dr. Baker?
Let's see. I can probably read them myself. Okay, what do we know about the immunogenicity of these mini binders in humans? Well, so there are now several designed proteins that are in humans in clinical trials. We don't have results yet, so we'll know soon. What we do know is that, for example, for the coronavirus binder, we've done experiments where mice were, the proteins were administered to mice every 3 days over a 3-week period, and then they were challenged with the virus following a final administration of drug, and there was absolutely no decrease in efficacy. So so we have not seen signs of drug neutralizing antibodies, but of course, with humans, we just don't know yet. And why that might be is because they're very small, very soluble, very stable. So they are probably a little bit harder to pick up by the immune system than most proteins. So are these binders orally absorbable? What I've described so far are probably not going to get across membranes very well, but I didn't talk about, we've designed a whole class of molecules which are somewhat smaller on the size range of cyclosporin, and these get across membranes very, very well, as well or better than cyclosporin. We actually have quite a large library of these macrocycles. They're cyclic compounds made out of both L and D amino acids, and so we can design these with very high precision. And so we're currently using this class of molecules to design inhibitors for intracellular targets. Let's see, could some of the neutralizing proteins function in non-human environments and neutralize pathogens in water, air? I think they could. It's just a matter of really how you're going to get them there. Like in water, you just mix them in. In air, you could make a mist, but obviously there's a large volume to cover. Let's see, have you in engineering reversible sensors? Let's see, so the sensors I described are in fact reversible, and that was the idea behind using them to detect the levels of neutralizing antibodies. So the idea is you have a sensor, which is in the off state, you add target, which is in this case, the receptor binding domain or the spike protein of the coronavirus, and that pulls the equilibrium of the sensor, which is dominantly in the closed state in the absence of target, it pulls it into the open state. Now, if you add something else which binds to the RBD, then it pulls the RBD away from the sensor and the sensor closes. So basically the whole concept of using this for vaccine monitoring is based on the reversibility of the sensor. And so we've shown these sensors are everything is governed by thermodynamic equilibrium. Let's see. Okay, how much computational power do you need to design and generate these structures de novo? Is AI part of the design and discovery process? Well, let's see. So I talked about different methods. The traditional Rosetta methods are pretty compute intensive. The deep learning methods turn out to be much more efficient. So for Rosetta structure prediction, which is what we've been doing for a long time, it's a Monte Carlo process where you're actually modeling the protein folding process. So the protein's folding up, sort of this physical model, you're searching for the lowest energy state. And because the landscape, there's so many possible states, we have to do many independent samples. Hundreds of thousands. So although each one just takes a minute or two, it adds up. For Rosetta TT fold, we run it on the server now. It's just a couple minutes, and it's not doing this large scale sampling. The network just outputs basically a single structure, which turns out to be quite good. So that's very much faster. Let's see. Yeah, so in terms of were we influenced by Eric Drexler's work? Yeah, I mean, so certainly the idea of making nanomachines is quite old, and that was something that Eric Drexler certainly has put a lot of, talked about a lot. And we're really excited that protein design methods are now at the stage where we can think about designing rotors and walkers and so forth. And I think it's going to be a really exciting next few years in this area. Let's see. Have you ever played with predictive folding in alternative environments, microgravity? No. Okay. How does Rosetta TT fold actually directly compare to TR Rosetta 2? It's quite a bit more accurate. And particularly, it's interesting. So we see that, you can see that in Cameo. If you just go there, you can compare results. We have a preprint up, and the paper will be out pretty soon. But where we really see a big difference is in molecular replacement. It's been kind of phenomenal. Like I said, I keep getting emails from people who've solved their structures by molecular replacement. And TR Rosetta was good, but there's just somehow the gap between TR Rosetta and Rosetta Fold is the threshold through which molecular replacement starts working. I already commented on the immunogenicity. Yeah, so what about, let's see, flexibility that will allow enzymatic activity? Yeah, so I didn't talk about it. We're working hard on designing catalysts, and they're thinking about how the substrate is going to get in and the product is going to get out become quite important. You want the right type of flexibility because you don't want the thing to be too squishy, because you won't be able to hold the catalytic residues in exactly the right place. What about the CNS access? Right, so we've also designed proteins that bind to the transferrin receptor, and those get across the blood-brain barrier in organ-on-a-chip models, and we have some preliminary data in animals that look good. So we're targeting now a variety of targets at the blood-brain barrier. We don't have any data about whether these compounds actually get into the brain, the ones that were the small proteins generally. We wouldn't really expect them to get across the blood-brain barrier unless they're fused to one of these shuttle proteins that we're designing. Oh, hi, Jeremy. Yeah, so what about end-link glycosylation? Yeah, so that's a good question. For the vanilla structure prediction, we aren't putting in end-link glycosylation, but for example, for modeling GPCRs, particularly the extracellular domains like the sweet receptor, it becomes really important to consider where the end-link glycosylation sites are. Also, on that note, the getting this group 2 influenza binder turned out to be was a real challenge because of a glycan that covers this groove that we were aiming for. And so a lot of the trick to getting that was figuring out exactly how to get in to avoid the glycan. Oh, hi, Jim. Yeah, I think that the sensors are totally genetically encoded, and we're actually trying to make sensors for RAS sensors and other types of sensors now. So I think you really could. I guess it would be fun to talk with you later about what markers you might actually want to detect. But
Can you possibly read the question for the rest of the audience?
Oh, sorry. Yeah, so Jim's asking, because the sensors are genetically, encoded, could you envision watching cells differentiate in real time with knowledge of specific markers? Yeah, I think it's a great idea. So, the sensors are completely genetically encodable. So you could have a real-time sensor of the expression of any particular marker you were interested in, and yeah, it'd be fun to talk about what markers would be interesting to do that with. Let's see. Has the result on the ACH on the trimer as an antiviral for neutralizations of variant of concern been published yet? Yeah, it's now, we put it on the archive, I think literally yesterday or the day before. So you can find that on bioRxiv now. Oh, hi, Bob. This is great. So Bob Stroud says, from the deep learning approach, can you say which of the input factors dominate prediction? How much weight is on inter-side chain pair distances, for example? Yeah, it's one of the things about these networks. They have tens of millions of parameters, so it's a little bit hard to figure out exactly what they're looking at. But surprisingly to me, we aren't really putting much weight on the details of the side chains aren't in there. It's really being modeled primarily at the level of the backbone. And so, they're recognizing patterns and sequences. It's just a little bit hard to say. One thing I can comment on, which is really quite fascinating, is when we do this hallucination approach, where we're just generating structures that the network thinks look like proteins, the things that it comes up with look kind of uncannily like the proteins that we've been designing, doing all, de novo designing over the years. They have nearly perfect secondary structural elements. They have like short loops. They don't have all the scraggly stuff that naturally occurring proteins generally have. And so it kind of shows that, like, traditional scientists staring at something, at a problem like protein structure design and protein folding, sort of come up with principles that you can write down on a piece of paper that then describe how to design proteins. And in fact, we've written papers that have titles like Principles for Designing Ideal Protein Structures. And then this network, you just sort of feed it all this data, and it kind of generates these ideal structures on its own. Let's see. Next question. If we were to hallucinate new enzymes around a catalytic site of interest, could we also optimize these enzymes for enhanced function with your approach? Let's see. I absolutely. And so that's one of the things we're doing now. You can basically take a functional region from a protein, either a binding protein or an enzyme or a metal binding site, fix that, and now hallucinate a protein around that. And so I think that's a very general approach. And then the network basically figures out the logical way to connect everything else. Okay, how much of this work is computational versus experimental? Trying to gauge what skills are needed to do this kind of work. Well, actually, it's a mix. And I have people come to my group who have very strong computational backgrounds and no experimental backgrounds, and vice versa. My talk was in a sense super misleading, because I showed you all these beautiful things of designs that work, but actually most designs don't work. And so there is a lot, like for the binders, we're making the designs on Agilent or Twist oligo chips. They're short proteins, so we can encode each one in an oligo, and then we're screening using yeast display for the ones that work. And so there's the success rate is very far from 100%. So coming up with experimental ways for identifying the proteins that work is important, as well as then characterizing them in detail is really important.
Can I ask a question, Dr. Baker?
Yeah.
You are leading this huge multidisciplinary team that does require expertise in all these really different areas. Can you describe your approach to managing? Like you're there at the top of this, directing this incredible ensemble.
I just have fun. I get bored. I get bored really easily. So the deep learning stuff has been so fun. I've gotten to learn a whole bunch of new things. So I mean, I just like learning new stuff, and I like interacting with people. And this is pretty much all I do. So I kind of, I think it's fun. And all these things, there's all this back and forth, we can develop some new deep learning structure prediction thing and then realize it can be turned around for protein design and then go apply it to coronavirus kind of in short order, because kind of all those things are going on. So a lot of my job is just make sure everyone's talking to each other all the time. Are we looking at this as a platform designing vaccines for other targets? For example, Neil's working hard on an HIV vaccine. So we think this will be a very powerful and general platform for design. In fact, Neil's universal flu vaccine just went into humans in clinical trials as well. And that's very exciting. That has multiple different hemagglutinins on the surface, and he's shown that elicits much broader immune responses. So do you expect protein binders can be designed for all human protein targets in the next 10 years? I think that really just depends on the resources that are put into it. There are some caveats. We currently are making designs based on proteins of known structure. We haven't done that using predicted structures yet. And there is a class of targets which is hard. Things that are very charged and very polar and don't have any hydrophobic character are hard to make design binders for. So the methods are still not perfect. Do you think there's a reason that nature has only sampled a small piece of possible amino acid combinations, or is it just a quirk of evolution? I think you could say it's a quirk, but evolution cares about function, and that's really what's important. And so why would nature go and explore some completely new area if it had a perfectly good solution accessible? And then remember how evolution works. It's sort of progressive, one or two mutations at a time. So it's going to really sample kind of around what it's already seen. And actually, that's a pretty good sampling strategy. As Francis Arnold has shown with directed evolution, I mean, it's incredibly powerful. I would say evolution hasn't sampled all structures because evolution didn't care about it or didn't need to. But now when we want to solve, 21st century challenges, being able to survey the whole space is a big advantage. Is there a method to predict whether a split protein will aggregate or not? Well, that's an interesting question. So I didn't try, but we've made an IL-2 mimic that binds, that's a very potent activator of IL-2, and we can split that, and and then we can target the two halves of the split well separately and only get reconstitution in where we want it. So far, that's worked pretty well with de novo design proteins, but we generally try a few different splits. So I'm not sure if it's going to be so predictable which ones work the best. So in nature, many proteins rely on metal ions. We have designed metal proteins with Rosetta, and that's something we're continuing to do with catalysis. How many protein variant spikes of COVID? Let's see, we probably tried against 4 or 5 strains now. It's a little bit hard to keep up with them. Do you think de novo design proteins will eventually replace protein therapeutics derived from the natural repertoire? Well, I think that's an interesting question. I mean, I think in the long run, as basically the way that reusing things that exist in nature is not really what we do in areas of technology outside of biology. Like we don't build buildings out of, we don't build buildings around trees, for example, except for decoration. So I think in the long run, as we learn more about design and questions about immunogenicity and so forth get worked out, that will become more and more the case. Are we open to collaborators getting in touch? Yes, we are very excited about collaborating. We have all these exciting methodologies we'd like to get out there. Let's see, I think I might be running out of time.
Yeah, you have to go at 2 on the dot?
Probably not. Okay, let's see. Could you expand on how these designer proteins are graded in the cell, proteasomal versus lysosomal degradation? Well, I think it's really going to depend on where they are. I mean, if they're just made in the cell, if they're getting endocytosed, we presume that they are getting into lysosome. What I didn't have a chance to describe is we designed proteins which undergo a large conformational change at low pH. They completely open up and they become membrane lytic. So we're trying to use those to solve the endosomal escape protein, basically the problem, basically to get proteins out of the endosome for nucleic acid delivery. Have we experimented with multivalent vaccines? Yeah, basically everything I described, those nanoparticles are highly multivalent. Oh, hi, Ava. Yeah, other than health-related problems, one of the things I'm really interested in is biomineralization. I mean, so you have things like bone and tooth and abalone shells, and like nature's figured out how to get calcium carbonate and calcium phosphate to mineralize. We're working hard to figure out how to do that with design proteins, because then you can make really interesting composite materials out of, semiconductors and other things. So that's one example. Then, some of those, like the Betabarrel channels, I think there's really a wide variety of filtering applications. So I think there's a lot of possibilities. We rely on people often coming to us and suggesting them, because of course we're not always worried know about the killer app. Yeah, so tell the story of a project from beginning to end would be a great talk. Yeah, well, you don't have time for everything. So yeah, well, those are great questions.
Well, thank you so much. It's absolutely fascinating. My eyes have been opened to an area I knew nothing about, and I know a lot of very high-level people were interested in your talk, Dr. Baker. We really appreciate you making the time to speak to our audience.
Okay, great. Well, it's fun to. I guess I get to see people in person, but it's fun to at least virtually. Okay, well, thanks. And if people would like to collaborate, send me emails. I mean, that's kind of the most fun. I've been telling people the key to doing great science is collaborating with great scientists. So yeah.
Okay, great. Thanks. Thank you.
Bye.
Bye.